Okay, so you sent over some research on these new techniques for editing insect genes.
Yeah.
And I have to say, like, wow, the possibilities are pretty mind-blowing.
It's really exciting stuff.
We're talking like pest control, disease prevention, even like revolutionizing agriculture.
Yeah, you know what's really interesting is that we're seeing a shift away from some of those older, more labor-intensive methods.
Yeah.
And these new techniques are opening up a whole new realm of possibility
I love that.
for researchers who might not have access to, fancy equipment
Ooh.
or like super specialized labs.
It's like democratizing science.
Yeah.
I love that. So what is the first technique that just like jumped out at you?
Well, there's this method called Deepa CRISPR,
Mm-hmm.
and it's surprisingly simple.
Deepa CRISPR.
Yeah,
Okay.
but so effective.
Break that down for me. How does that work?
So basically, you're injecting a mix that contains Cas9,
Uh-huh.
which is an enzyme that can cut DNA,
Right.
and a guide RNA that tells the Cas9 exactly where to cut. And you inject that directly into the body cavity of an adult female insect.
Got it.
So this mix is called Cas9 ribonucleoproteins, or
Okay.
Cas9 RNPs
Cas9 RMPs.
for short. And these RNPs make their way to the developing eggs.
So you're basically bypassing all that like embryo micro injection.
You got it.
That's really clever, but does it work?
It does.
Okay.
Yeah, researchers have successfully used Deepa CRISPR
Wow.
to induce mutations in both cockroaches, specifically the German cockroach,
Okay.
which is Blatella germanica,
Volatilization.
and red flower beetles, which is Tribolium castaneum.
Okay.
And one of the studies I was looking at, they actually had a picture of a cockroach
Ah.
with these like mosaic patterned eyes because of the gene editing.
Wait, I got to see this.
Yeah.
Hold on.
It's pretty wild.
Whoa, those eyes are wild. It's like a living piece of abstract art.
Yeah, it's pretty cool.
But more importantly, it's proof that this Deepa CRISPR is like really changing the genes.
Exactly. And what's interesting is that they found Deepa CRISPR to be more efficient in species with continuous reproduction,
Okay.
like those cockroaches.
So like they're constantly reproducing.
Yeah, because there's more opportunity for the Cas9 RNPs to get incorporated into the egg's DNA
Okay.
during a process called vitellogenesis.
Vitellogenesis.
Yeah.
That rings a bell.
It has something to do with yolk production.
Yolk production, yeah.
Yeah, so vitellogenesis is when yolk proteins are produced
Okay.
and deposited into the developing eggs.
Got it.
And so in species that are constantly producing eggs,
Uh-huh.
there's a larger window of opportunity for those gene editing molecules to work their magic.
So continuous reproduction equals more chances to hit that genetic sweet spot.
Yeah.
Okay, but what about insects that don't reproduce continuously?
Right.
Like those beetles you mentioned.
Yeah. So they found that even in the beetles,
Okay.
which have discontinuous reproduction,
Uh-huh.
they could still achieve successful gene knock-in using Deepa CRISPR.
Yeah.
And they did this by introducing single-stranded DNA fragments
Okay.
called SODNs alongside the Cas9 RNPs.
Got it.
And these SODNs essentially serve as templates for the desired genetic changes.
So even if that window of opportunity is smaller,
Yeah,
you can still kind of sneak those edits in.
exactly.
Pretty impressive. But I imagine there are some limitations to this technique.
Right. It's not a magic bullet for every insect species out there.
Right.
The researchers point out that Deepa CRISPR might not be equally effective
Yeah.
in all insects.
Makes sense.
Factors like differences in reproductive cycles
Sure.
and the specifics of vitellogenesis could make it more challenging to
Yeah.
apply in some species.
Uh-huh.
So they've given us a roadmap for future research, basically,
Right.
highlighting those areas where we need more investigation.
Well, that's responsible science, right?
Yeah,
Acknowledging the limitations while also celebrating the success.
exactly.
Speaking of successes,
Right.
you mentioned another technique,
Yes.
remote control.
Remote control.
What makes this one so special?
So remote control is all about working with nature, not against it.
Okay.
So it utilizes the natural process of vitellogenesis
Sorry.
to deliver those Cas9 RMTs right into the insect ovaries.
I'm intrigued. Tell me how they do that.
So they use something called peptide ligands,
Okay.
which are like little molecular keys
Uh-huh.
that fit into specific locks on the surface of cells.
Okay.
And in this case, the locks are receptors involved in yolk protein uptake.
Got it.
And the keys are derived from the yolk protein precursors themselves.
So it's like they're disguising those gene editing molecules as something that the cells naturally want to take in.
Yeah.
Clever. And what kind of insects have they tested this on?
So remote control has a broader application
Oh.
than Deepa CRISPR.
Okay.
It's proven successful in a variety of insects: mosquitoes, beetles, wasps, even ticks.
Wow.
So they've used it on 3 different kinds of mosquitoes.
Uh-huh.
Aedes aegypti,
Okay.
Anopheles stevensii,
Anopheles.
and Culex pipiens pallens.
Okay.
And there's also been successful trials with the red flower beetle Tribolium castaneum,
Right.
the parasitic wasp Nasonia vitripennis,
Right.
the Asian citrus Psyllid diaphragma citri,
Got it.
and even the black-leg tick Ixodes scapularis.
That is quite a lineup.
It is.
And are they all using the same key?
So in many of the studies, yes.
Okay.
The ligand they use is called P2C,
P2C.
and it's derived from a yolk protein
Okay.
found in the fruit fly, Drosophila melanogaster.
Drosophila.
And it's surprising how well this P2C works across different species.
Right.
They usually combine P2C with the Cas9 RNPs,
Okay.
along with some helper molecules that allow the RNPs to escape from little compartments inside the cell called endosomes.
Wait, endosomes, can you back up and just explain what those are?
Yeah, so when those P2C Cas9 RNP complexes
Okay.
enter the cells in the ovary,
Uh-huh.
they get wrapped up in these tiny membrane-bound sacs
Okay.
called endosomes.
Got it.
And it's part of the cell's normal process.
That-
Right.
Of sorting and transporting molecules.
Okay.
But for the Cas9 RNPs to reach the DNA
Yeah.
and make those edits,
Right.
they need to break free from the endosomes
Okay.
and reach the nucleus.
So they're like trapped in these molecular bubbles.
Exactly.
How do they escape?
So that's where those helper molecules come in.
Okay.
They're called endosomal escape reagents,
Okay.
or EARs,
Yeah.
and they essentially poke holes in the endosomes,
Uh-huh.
allowing the Cas9 RNPs to escape and get to work.
Okay, so it's like a very elaborate escape plan.
It is.
Okay, so how effective is this whole remote control scheme? Are we talking like significant gene editing?
The results are pretty impressive.
Okay.
One of the studies that I came across
Yeah.
involved using remote control on silkworms
Oh.
to create mutants with this like oily skin phenotype.
Okay, silkworms.
Yeah.
That's oddly specific, but hey, it's proof of concept, right?
It is exactly, but there are some caveats.
Okay.
While that PDC ligand has been surprisingly versatile.
Its effectiveness does vary
Okay.
across different insect species.
Right.
So in some cases, like with the whitefly,
Amesia tabaci.
Phomesia tabaci, researchers actually had to use a species-specific yolk protein ligand
Got it.
for remote control to work properly.
So it's like they have this master key ring
Yeah.
with a bunch of slightly different keys.
Yeah.
And you might need to try a few to find the one that fits.
Exactly.
Okay, so there's still some fine-tuning to be done. What else should we keep in mind about remote control? Anything else interesting in the research that kind of stood out?
So one thing that stood out to me was the debate surrounding the use of those endosomal escape regions.
Oh, really?
So some studies have shown successful gene editing even without using ERs.
Interesting.
So this suggests that, at least in some insect species,
Oh.
the cell's natural process of taking in molecules, called endocytosis,
Right.
might be sufficient to get those Cas9 RNPs where they need to go.
So it could simplify the whole process.
Exactly.
Less is more.
Yeah, and I imagine more research is needed to figure out which insects might be good candidates for
Yeah.
this simplified approach.
Makes sense, but it opens up some possibilities.
Absolutely.
Speaking of learning, you mentioned another technique
Yeah,
that piqued your interest.
right.
BAPC.
BAPC.
What's the story with that one?
So BAPC stands for branched amphiphilic peptide capsules.
Okay.
Think of them as tiny self-assembling capsules
Yeah.
made from peptides,
Okay.
those short chains of amino acids that are the building blocks of proteins.
Right.
And these capsules can encapsulate the Cas9 RNPs,
Got it.
protect them, and deliver them into the cells.
So another clever delivery system.
Is.
Has this been tested in any insects?
Yes, and with some success.
Okay.
Researchers have used BAPCs to deliver Cas9 RNPs into the parasitic wasp,
Okay.
Nasonia vitripennis,
Nasonia vitripennis.
and they were able to induce mutations in a gene that affects eye pigmentation.
Parasitic wasps.
Mm,
They're both fascinating and kind of creepy.
they are.
The fact that they got this technique to work in such a complex insect is pretty impressive.
It is, and what's particularly exciting about BAPC
Yeah.
is its potential as an alternative to embryo microinjection.
Okay.
Remember all those challenges we talked about with the
Right.
small egg size
Right.
and the specialized equipment?
Yeah.
BAPC could bypass all those hurdles,
Right.
making gene editing more accessible
Wow.
to researchers who don't have access to those fancy setups.
There's the democratization of science again. I'm all for that.
Me too.
So we've got these three Pretty amazing techniques.
Yeah.
DEPA CRISPR,
Right.
remote control,
Yes.
and BAPC.
BAPC.
Each one with its own, unique approach to insect transgenesis.
Yeah.
Before we move on, are there any, overarching challenges or trends that you've noticed across these different approaches?
So one thing that stands out is that researchers are constantly pushing the boundaries of precision
Yeah.
and efficiency.
Okay.
They're fine-tuning these techniques, optimizing parameters, and testing new combinations of molecules
Right.
to achieve the most targeted gene editing possible.
It's like they're refining their genetic sculpting tools,
Yeah,
making them sharper and more precise with each new discovery.
that's a great way to put it.
So we're just scratching the surface here.
I think so.
I can't wait to see what they come up with next. But before we get too far ahead of ourselves, let's take a break and come back to explore the potential applications of these groundbreaking techniques.
This is good.
Seen a few. Yeah. So we've covered some pretty mind-blowing techniques for, editing these insect genes.
Yeah.
But what does this all mean for the real world? What kind of impact are we talking about here?
The potential applications are vast.
Oh,
So imagine targeting specific genes in pest insects to disrupt their reproduction
okay.
or make them more susceptible to existing insecticides.
Right.
We could develop new pest control strategies that are more environmentally friendly and sustainable.
So instead of, blanketing crops with these broad-spectrum pesticides,
Right.
that harm beneficial insects and pollute ecosystems, we could have these, targeted solutions that address, very specific pest problems.
Exactly.
That sounds like a huge win for, agriculture and the environment.
Yeah, for sure. And then there's the potential for disease prevention.
Okay.
Mosquitoes, ticks, and other disease-carrying insects,
Right.
they're a major global health concern.
Yeah.
With gene editing, we could potentially prevent these insects from transmitting diseases like malaria, dengue fever, Lyme disease.
Yeah, that's an exciting thought, like a world without mosquito nets and constant worries about tick bites.
Yeah.
But how would that actually work?
So there's a few approaches.
Okay.
We could engineer mosquitoes that are resistant to the parasites that cause malaria,
Okay.
or modify their genes so they can't transmit the viruses that cause dengue.
Right.
In the case of ticks, we might be able to target genes that make them less likely to bite humans
Okay.
or less capable of carrying the bacteria that causes Lyme disease.
Wow, that's incredible. It's like science fiction becoming reality.
It is.
But I imagine there are still some hurdles to overcome before we see these applications in the real world.
Of course, safety is paramount
Right.
before releasing any genetically modified insects into the environment.
Yeah,
We need to conduct rigorous testing
of course.
to make sure that they don't have any unintended negative effects on the ecosystems or human health.
Makes sense. And what about public acceptance? I feel like people tend to be wary of genetically modified organisms,
Right.
especially
Right.
when it comes to insects.
Yeah.
So how do we address those concerns?
I think transparency and communication are key.
Okay.
We need to engage in open and honest conversations with the public about the potential benefits and risks of these technologies.
Right.
It's about building trust and understanding.
Yeah, I think that's so important. Science shouldn't happen in a vacuum.
Yeah,
It needs to be part of, this larger societal conversation.
for sure.
Now, you mentioned that these gene editing techniques
Yeah.
could also have applications beyond pest control and disease prevention.
Right.
What else are we talking about here?
So think about agriculture.
Okay.
We could engineer bees that are more efficient pollinators,
Oh, wow.
or develop silkworms that produce stronger and more versatile silk.
Okay.
We could even explore ways to use insects as sustainable sources of protein for food or animal feed.
That's fascinating. It's like we're moving from this adversarial relationship with insects
Right.
to one of collaboration and mutual benefit.
Exactly.
I like that vision.
Yeah, me too. But as with any powerful technology,
Right.
we need to proceed with caution and a deep sense of responsibility.
Yeah.
We need to consider the ethical implications,
Uh-huh.
assess the risks,
Right.
and ensure that these advancements are used for the benefit of humanity and the planet as a whole.
Well said. So we've got the potential to solve some of the world's most pressing problems
Yeah,
with these gene editing techniques.
it's pretty amazing.
But I'm also curious about what they can teach us about the insects themselves.
Right.
What are we learning about the inner workings of these fascinating creatures?
These gene editing tools are like powerful lenses
Okay.
that allow us to zoom in on the intricate machinery of life.
Yeah.
We can now pinpoint specific genes
Uh-huh.
and see how they influence an insect's development
Okay.
behavior,
Right.
and physiology.
So it's like we're cracking the code of insect life.
Yeah.
What kind of insights are we gaining from this?
So we're learning about the genes that control wing patterns in butterflies.
Oh, wow.
The genes that govern social behavior in ants.
Okay.
The genes that determine an insect's resistance to pesticides.
Interesting.
A whole new world of discovery.
That's so exciting. It feels like we're on the cusp of like a new era in insect biology.
I think so.
But I'm also aware that science is rarely like a straight path to success.
Right.
What kind of challenges are researchers facing as they explore this uncharted territory?
Well, one challenge is the sheer diversity of the insect world,
Okay.
with over 1 million known species, each with its unique biology and genetics.
Right.
It's really difficult to develop gene editing techniques
Yeah.
that work universally across all insects.
So it's like trying to find a one-size-fits-all solution
Yeah.
for this group of creatures that are anything but uniform.
Exactly.
That makes sense.
Yeah.
What other obstacles are they encountering?
Another challenge is the complexity of insect genomes.
Yeah.
Some insect genomes are even larger and more complex than the human genome.
Really?
Finding and targeting specific genes within these vast genetic landscapes can be a daunting task.
It sounds like navigating a labyrinth.
It is.
But I imagine researchers are developing new tools and strategies to meet these challenges.
Yeah, they are.
Right.
They're using advanced computational methods to analyze those complex genomes,
Okay.
identify promising gene targets,
Right.
and predict potential off-target effects.
So it's this like back and forth between this
Yeah.
incredible complexity of insect biology
Right.
and the ingenuity of human researchers.
It's a constant process of refinement and innovation.
That's what makes science so fascinating.
It is.
Now, I'm eager to hear your thoughts on the future of insect transgenesis.
Okay.
What do you see on the horizon for this field?
I think we're just beginning to scratch the surface of what's possible.
Yeah.
As gene editing techniques become more precise, efficient, and accessible,
Right.
we'll see an explosion of new discoveries and applications.
Okay.
We'll develop new strategies for pest control, disease prevention,
Huh?
create innovative biomaterials inspired by insects,
Wow.
and gain a deeper understanding of the evolutionary processes that have shaped this incredible diversity of life.
It's an exciting time to be involved in this field.
It is.
And an exciting time to be learning about it.
Yes.
Now, before we get too carried away with all the possibilities,
Right.
I think it's important to touch on the potential risks
Yeah.
associated with these technologies.
Sure.
We've already talked about the need for rigorous testing and public engagement,
Mm-hmm.
but are there any other concerns that we should be aware of?
So one concern is the potential for unintended ecological consequences.
Okay.
Even with the best intentions,
Right.
releasing genetically modified insects into the environment
Nah.
could disrupt delicate ecosystems.
It's that butterfly effect scenario.
Yeah.
You tweak one thing,
Right.
and it sets up like this chain reaction with unpredictable results.
Exactly. Ecosystems are incredibly complex and interconnected.
Right.
It's difficult to predict how a change in one species, even a seemingly minor one, will ripple through the entire system.
Makes you realize how much we still don't know about the intricate web of life on Earth. Are there any specific examples that come to mind where things could go wrong?
So think about those gene drives that we talked about earlier.
Okay.
They have the potential to spread a desired gene through an entire population of insects incredibly quickly.
Right.
While this could be incredibly powerful for controlling invasive species or disease vectors,
Yeah.
and there's also the risk of those genes jumping to other species or spreading beyond the intended target area.
So it's like unleashing a genetic wildfire that could burn out of control.
Yeah.
That's a sobering thought.
It is, and it highlights the need for robust safeguards and containment strategies.
Right.
Researchers are exploring ways to engineer gene drives with built-in kill switches.
Okay.
Or to limit their spread to specific geographical areas.
Got it.
But these technologies are still in their early stages,
Right.
and there's a lot more work to be done
Yeah.
before we can confidently deploy them in the real world.
It seems like the scientific community is very aware of these potential risks and is taking steps to address them.
Yeah,
But it also sounds like there are a lot of unknowns.
right.
So how do we balance the potential benefits of these technologies
Yeah,
with the need to proceed cautiously and responsibly?
that's the million-dollar question.
Yeah.
There's no easy answer.
Right.
It requires a multifaceted approach
Okay.
that involves open communication, rigorous risk assessment, and ongoing monitoring.
So we need to involve not just scientists, but also like ethicists, policymakers, and the public in these conversations.
Absolutely. It's a shared responsibility.
It makes you realize that science isn't just about, like, technical advancements. It's also about values, ethics, and societal choices.
Right.
We can't just focus on what's possible.
Yeah.
We also have to consider what's desirable and what's right.
I couldn't agree more. Science is a powerful tool,
Yeah.
but it's up to us to decide how we use it.
Right.
We need to ensure that these technologies are developed and deployed in a way that benefits humanity and the planet as a whole.
So as we wrap up our exploration of these, like, groundbreaking techniques,
Yeah.
what's the one key message you want our listeners to take away from this deep dive?
I want them to remember that insect transgenesis is a field with immense potential,
Okay.
but it's essential that we proceed with caution, responsibility, and a deep respect for the interconnectedness of life on Earth.
Yeah.
These are powerful tools that can be used for good or for ill.
And it's up to us to ensure that they're used wisely.
That's a powerful message. And it's a reminder that science isn't just about discovering new knowledge. It's about using that knowledge to create a better future. For all, thanks for joining us on the deep dive. So we've talked about this incredible potential of these new insect transgenesis techniques.
Right.
But you just mentioned this other side to the coin.
Right.
These potential risks.
Yeah.
Ethical dilemmas. It seems like every time we take like this giant leap forward in science,
Yeah.
we have to grapple with this whole new set of challenges.
That's the nature of progress, isn't it? It's like opening a door to a new room,
Right.
only to discover it leads to a hallway with even more doors to explore.
And sometimes those doors lead to unexpected or even like unsettling discoveries.
Yeah.
What are some of the specific concerns that you think deserve the most attention as we like venture further into this uncharted territory?
Well, I think it's worth emphasizing those ecological risks,
Okay.
even with, like, the best intentions.
Releasing these genetically modified insects into the environment could have like unforeseen consequences.
Right. It's like that saying, the road to hell is paved with good intentions.
Yeah.
We could be trying to solve one problem, like malaria transmission,
Right.
but end up creating a whole new set of problems.
Exactly. Ecosystems are incredibly complex
Right.
and interconnected, and it's so difficult to predict how like a change in one species,
Yeah.
even a seemingly minor one,
Right.
will ripple through that entire system.
It makes you realize how much we still don't know
Yeah,
about this, like, intricate web of life on Earth.
a lot of unknowns.
Are there any, like, specific examples that come to mind where things could go wrong?
So, those gene drives we talked about earlier,
Yeah.
they have the potential to spread a desired gene through an entire population
Yeah.
incredibly quickly.
Right.
So while this could be like incredibly powerful for controlling invasive species or disease vectors,
Right.
there's also that risk of those genes jumping to other species. Okay.
Or spreading beyond, like, that intended target area.
So it's like unleashing this genetic wildfire
Yeah.
that could burn out of control.
Exactly.
That's a sobering thought.
It is, and it highlights the need for, like, robust safeguards and containment strategies.
Right.
So researchers are exploring ways to engineer gene drives with built-in kill switches,
Okay.
or to limit their spread to specific geographical areas.
That makes sense.
But these technologies are still in their early stages, and there's a lot more work to be done
Right.
before we can confidently deploy them in the real world.
It seems like the scientific community is very aware of these potential risks
Yeah, for sure.
and is taking steps to like address them.
Yeah.
But it also sounds like there are a lot of unknowns.
There are a lot of unknowns.
So how do we balance the potential benefits of these technologies with the need to proceed cautiously and responsibly?
That's the million-dollar question, isn't it? And there's no easy answer.
Right. It requires a multifaceted approach that involves open communication, rigorous risk assessment, and ongoing monitoring.
So we need to involve not just scientists, but also ethicists, policymakers, and the public
Yeah.
in these conversations.
Absolutely. It's a shared responsibility.
It makes you realize that science isn't just about technical advancements. It's also about values, ethics, societal choices.
It is for sure.
We can't just focus on what's possible. We also have to consider what's desirable and what's right.
I couldn't agree more. Science is a powerful tool.
Yeah.
But it's up to us to decide how we use it.
Right.
We need to ensure that these technologies are developed and deployed in a way that benefits humanity and the planet as a whole.
So as we conclude our deep dive into this fascinating world of insect transgenesis,
Yeah.
what's like the one key takeaway?
I think the most important thing to remember is that this is a field with immense potential, but it's crucial that we proceed with a sense of humility, responsibility, and a deep respect for that delicate balance of life on Earth. These technologies offer these incredible opportunities
Yeah.
to solve like these pressing global challenges. But they also come with inherent risks
Right.
and ethical dilemmas.
Yeah.
It's up to all of us to engage in thoughtful dialogue, consider diverse perspectives, and work together to shape a future where these technologies are used for the benefit of all.
That's a powerful and thought-provoking message. Thank you for sharing your expertise and insights with us today. And to our listeners, thank you for joining us on the Deep Dive. We hope this episode has sparked your curiosity and inspired you to delve deeper into this fascinating world of insect transgenesis.